Hi guys, so this is the last minute revision video for biomolecules for the HSC 2020 chemistry exam, which is going to be scheduled in a couple of days, right? So now let's see what exactly are the things that you're going to study. Now biomolecules is a very bigger topic and it has a lot of structure, so students mainly are not comfortable with this topic. But the most important thing is actually it is one of the easiest topic, provided that you have a grip of chemistry, the basics, and you love you love how to write the structures of these complex biomolecules. But the weightage of this particular topic is very less, so students tend to ignore this topic or actually keep it in option. But actually it's not worth keeping this in option because it's one of the easiest topic. Okay, so here are the last minute revision tips that you should follow when you're preparing for this. So the type of questions which have generally appeared in the paper is basic MCQs. They are the factual types. They may ask you the linkage in DNA molecule or linkage of like base sugar and phosphoric acid, phosphodiester linkage at what positions and so on. And then objective questions like very simple one, structures of disaccharides or any one of the polysaccharide has already been asked, right? So they are not difficult provided that you know how to handle those structures. Details on proteins and amino acids. Now proteins and amino acids have been one of the most important parts, right? So try to do this very nicely and very thoroughly. Okay, so let's see what others you're going to do in separate sections. In carbohydrate, the structure of disaccharides like galactose, lactose, maltose, and the polysaccharide like amylose, amylopectin, right? And then cellulose and so on. The linkages normally in the case of in the case of sucrose, it is C1, C2, right? Whereas in the case of other sugars, it is C1, C4. Remember, except one of the branched one. Okay. So then polysaccharides, as I've already said, try to remember the linkages and the structure. Anomeric carbon in each sugar. Normally, it is the carbon. Which contains. It is a carbon of the aldehyde or the ketonic group. For example, in the case of what do you say, glucose. Anomeric carbon is C1 because C1 is aldehyde. In the case of fructose, it is C2 because C2 is a ketonic group, right? So basic MCQs regarding that. Reducing, non-reducing sugar. All sugars except sucrose are reducing sugar. Sucrose is not reducing because anomeric carbon atoms are linked, right? And you should remember that what exactly do you mean by a reducing and non-reducing sugar? If the anomeric carbon atoms are linked, right, they are basically non-reducing sugar, the others are reducing sugar. And the preparation like using hydrolysis and so on, just a very simple method that you should do it, right? And uses as well. Okay, now the next part, proteins and amino acids. Structure of a protein, not the complex structure, but it is long chain alpha amino acids, right? Long chain alpha amino acids and peptides, right? A dipeptide contains single peptide linkage, tripeptide contains two peptide linkage, and so on. Binding forces like primary structure of protein, secondary, tertiary. What are the binding forces? Hydrogen bonding, peptide linkage, van der Waals forces, disulfide linkages, right? These could come as the MCQs. And the isoelectric point, the pH at which the zwitter ion exists. What exactly is a zwitter ion? It's basically the neutral bipolar ion which is formed as a result of proton transfer from acid to base, right? So this is one that you should remember. And then enzymes and hormones, elementary idea, what are enzymes, what are hormones, what are the properties and different types, right? And then hormones, reproductive, non-reproductive, progesterone, estrogen, and then the other types of hormones, secretions of pituitary glands, very basic elementary ideas, right? And then you have lipids, again the basic ideas, what are oils, what are fats, they are triglycerides or of, so the thing you have already know. Oils are triglycerides of glycerol and unsaturated fatty acid. When it is saturated fatty acids, you call it as fat. Okay, and then there are different classifications: sphingolipids, glycolipids, and so on. Lipoprotein, and their use is very small section. So even I mean, this is a very small section. So if it's possible, you could do it. Now vitamins, very simple elementary idea. The different types of vitamins like water soluble, sorry, it should be okay. it should be fat soluble. Okay, this is just a printing error, so it should be fat soluble, right? And then classification, and on the basis of the structure, sources of the different vitamins, and most important thing, the series of vitamin B. You have vitamin B1, then you have B2, then you have B3, then you have B6, sorry, B5 is there, and then you have B6 and B12. B12 is important particularly because it consists of a transition element which is cobalt. Okay, so this is, and now comes the most important part, that is nucleic acids. Nucleic acids, the linkages are very important. Bases connected to sugar, what carbons, and then two phosphoric acids are linked at what carbons, right? It's a phosphodiester linkage. Sugar in DNA is 2-deoxy D-ribose sugar. In RNA, it is D-ribose only. Oxygen is removed from which carbon in DNA, right? In DNA, sugar, right? Base sugar linkages I have already discussed, and replication, the elementary ideas, but yes, this is very, very important, right? So this is very, very important, and it could form, I mean, I would say that a lot of questions are expected from this particular topic, because this is not just important, but also you could say that a lot of data is already there that is pretty much relevant, right? So this was about the section on biomolecules. Now, I know that it's pretty much difficult for you to start revising the structures if you haven't done throughout the year, but at least you could go through the structure and try to understand it, right? It's not a complicated topic, as I've already said, right? And the major section is carbohydrates, proteins, and nucleic acids. This you should do thoroughly, right? And of course, the bases like purines, pyrimidine, if you could do the structure, it's very good. I think last year it was asked, right? So I wish everyone very good luck studying this particular topic. Stay tuned for other topics, last minute revision videos, which would be released in a couple of hours. Thank you.